Hello everyone, I am Azira Nafisah binti Amiruddin with metric number A165108. So today I will be presenting my video seminar project for STBC3022 Genomics. Clostridium difficile is an anaerobic gram-positive and spore-forming bacterium that is responsible for infections ranging from mild diarrhea to pseudomembranous colitis. The infection most commonly affects people or elderly patients who have recently been treated with antibiotics such as fluoroquinolones and cephalosporins. To make matters worse, this infection can spread easily to others. Besides, Clostridium difficile is known as North American pulse field type 1, restrictions endonuclease analysis type BI or PCR Ribotype 027. So this is because in 2003 this bacterium has caused a large outbreak in North America. Since then, cases have been reported worldwide such as in Europe and Australia. Nevertheless, cases of NAP1 infections can rarely occur in Asia, but this infection has been already described in Hong Kong and Guangzhou, China. NAP1 is characterized by an in vitro overproduction of toxins A and B as well as a binary toxin production that is most probably due to the deletions of 18 base pairs in TCDC gene. In the present study, the first NAP1 outbreak has been reported in hospital in mainland China. Moving on to methodology, the pathogen diagnosis was conducted by taking stool sample from a CDI case patient to test for its toxin using enzyme immunoassay and CCF-agar. Next, epidemiological investigations was also conducted by collecting clinical information from medical records. By using these retrospective case searches, it's been reported that 75 patients have diarrhea, while 20 of them were diagnosed as CDI, including one patient with BSI. Besides, the interview with clinicians, nurses, and nurse assistants were also conducted along with collecting architectural structure of the ward and room management. After that, validation and typing the strains into outbreaks were conducted by using PCR and MLST. Last but not least, a 500 base pair library was constructed and a whole genome sequencing was performed using Illumina High Sequence 2000 platform in order to obtain a 100 base pair paired and read. Along with that, reconstructions of phylogenetic tree as well as detections of genomic single nucleotide polymorphism was also conducted in order to obtain the transmission routes of the outbreak. The result of this study started off with the identification of BSI case in one patient referred as patient 13 who was initially admitted due to pneumonia. But after 7 days, he developed CDI symptoms such as fever and diarrhea for several days and repeatedly admitted in hospitals. He was then treated by vancomycin as it is believed that this antibiotic works best with CDI. These situations had urged the researchers to do some investigations regarding this matter. By conducting retrospective study of clinical information of patients with CDI in the ward, there were about 90 patients were identified from March 2012 to April 2014. Moreover, most of the patients are in the range of 65 years old and above whom had been received antimicrobial treatment before diarrhea. The antibiotic that has commonly used in this ward to treat CDI was third-generation cephalosporins and fluoroquinolones. According to few studies, this highly toxic strain of Clostridium difficile has already resistant to fluoroquinolone antibiotics. From the laboratory study, the isolates were characterized into 5 sequence types by using MLST. ST1 strain had infected 11 patients, meanwhile ST37 and ST81 had infected 1 patient respectively. The genome sequences provide the detailed molecular evidence to connect part of the transmission route. The first one is the phylogenetic analysis. It showed that all NAP1 isolates in this ward belong to the same clone which existed in the ward in 3 years and caused 2 outbreaks. The last but not least, whole genome sequencing data validate that the strains in 2 outbreaks in consecutive years were only different by 2 stable SNPs. This indicates that the infected strain may exist in the hospital and transfer again in the following year. So this is the first investigating outbreaks of healthcare-associated Clostridium difficile and AP1 strain infection in China, and the researchers had reconstructed the transmission map by combining retrospective epidemiological analysis with whole genome sequencing. The question is why there is no report of CDI outbreaks in China being published. The first problem is very few hospitals in China can perform Clostridium difficile culture, which may result in many undetected CDI cases. Secondly, the lack of anaerobic laboratory equipment and limited CDI knowledge by healthcare staff also contributes to this problem. Furthermore, there is no reference center for typing Clostridium difficile strains in China and identifications of different PCR ribotypes. And it's been said that whole genome sequencing has helped in boosting Clostridium difficile research, also enriching the HAI pathogen database, as well as HAI outbreak tracing clues. Last but not least, WGS also improves the completion of Clostridium difficile in AP1 in global epidemiology distribution. In conclusion, the molecular epidemiology based on whole genome sequencing should be recommended for HAI investigations in addition to conventional field epidemiology investigation. And based on their genetic diversity, we can assume that the toilet, bathroom, and janitors equipment room may be contaminated area that may suggest it to improve infection control measures in the following healthcare. And with that, thank you for your attention.